Hi everyone, this corresponds with the first lecture posted during our week of transition in which we were switching from face-to-face classes to online instruction due to the COVID-19 pandemic. So appropriately, this lecture is about spreading and causing disease. This is looking at overall how we characterize diseases based on their transmission and epidemiology, studying epidemics. So this is looking at kind of a population and abstract level. The second part of this lecture, which was posted separately, is kind of about pathogen and disease progress within a single host. We'll be focusing on defining epidemiology and thinking about the ways in which we quantify it. There's different metrics here and different ways of thinking about this transmission and the severity of it. So we'll distinguish between morbidity and mortality, as well as between incidence and prevalence. We'll also look at different patterns of incidence, so whether the disease is sporadic, endemic, or epidemic, and what we mean by those terms. We've talked about Koch's postulates before, but we'll think about their importance in identifying what we call etiological agents. We'll also characterize diseases based on the evidence we have about them, their signs, symptoms, and syndromes, as well as the rate of spread, so whether it's communicable, contagious, or non-communicable. We'll also think about patterns of transmission, distinguish between different types of reservoirs and modes of transmission, and track the progress of an infection to define different phases. So one image that I wanted to show you is this really interesting visualization. If you take a moment to look at it, in class when we do this, I usually have students kind of talk to their partner about what this image is showing. So maybe take about 10 seconds, stare at it, and think about what this might be. So as you're looking at this, you might start to see some familiar shapes. For example, this one right here might look a bit like Africa. This might look like the United States and Mexico down here, as well as South America. Over here we can see India and Australia down here. So what this, and even Alaska up here. So what this graph is, or not graph, but what this image is showing us is actually a world map that is visualized just using flight patterns. So for any given day or time period, kind of the number of flights that are happening, and we can start to see country borders, we can see continent borders, so we start to see all these different patterns that emerge just from our movement on a global level. So when we're thinking about the spread of COVID-19 and comparing it to other coronaviruses like SARS and MERS were pretty isolated where they emerged. COVID-19, SARS-CoV-2 has spread throughout the entire world. And when we look at a map like this, we can see why. If something is very contagious and it sticks around for a long time and we are constantly moving, constantly shipping different supplies, constantly moving people, it presents a lot of opportunities to spread a pathogen or a contagion. So we're going to be thinking about kind of measuring this movement and measuring the disease progress in different populations throughout the course of this lecture. So if we're thinking about defining first and then quantifying epidemiology, first thing we have to define is epidemiology. So that relies on knowing what an epidemic is. We kind of as we understood that COVID-19 was spreading throughout the world, there were people talking about it being an epidemic and then a pandemic. An epidemic is the rapid spread of infectious disease to a lot of people in a population within a very short period of time. So that was clearly happening early on. It was just a matter of whether this was a global epidemic. That's kind of the definition of a pandemic. So when we're studying these epidemics, what we're doing is epidemiology. It's the study of the distribution and timing of infectious disease occurrences and how they are transmitted and maintained in nature. So how does a pathogen spread? What's the rate at which it's spreading? Who is it affecting? What proportion of the population? And how is that sustained, if it is sustained at all? So there's an inherent relationship between understanding what an epidemic is and the study of epidemiology. When we're thinking about epidemiology, our understanding of the world is only as good as our models and, more importantly, the data that we put into it. And epidemiology helps us get a more accurate understanding of disease transmission and the way in which different people, different organisms, different pathogens interact. So this image was published in the American Journal of Medicine in 1984, pretty early on in the AIDS crisis, and it's based on what we call an index case. So that's kind of like the first person, patient zero, to have a particular pathogen. This man, Gaten Dugas, was assumed to be patient zero. They looked at kind of his sexual interactions with other people and tried to track the spread of the HIV virus and AIDS. Remember that AIDS is the syndrome that happens when you have a very high viral load of HIV. HIV is the causative agent. It's a retrovirus. That's the actual pathogen. So this transmission map really puts the blame very squarely on Gaten Dugas, which is maybe not accurate. And so as we learn more about HIV, about its genetics, and about how it spreads, we can start to put together much more accurate transmission maps. So this map was an updated early transmission of map of HIV, which was published in Nature magazine in 2016. So you can see this is a little bit more nuanced. It has a little bit more about the taxonomy of the virus, how it mutated, and more accurate metrics of how it spread without really putting the blame solely on one person. So when we're looking at a widespread epidemic, this is an important thing to take into consideration, especially one that has a lot of stigma. If we're thinking about patients spreading something like COVID-19, we might want to have a transmission map like the one on the previous slide. There's been a lot of kind of shady stuff happening with phone security and personal privacy. For example, if we have a large company or a government that is tracking GPS data on our phones and someone tests positive for COVID-19, then by looking at kind of their GPS history, we can see who they came into contact with. So physical proximity or actual relationships and exchanging of fluids and different things like that. So there's been incidents in other countries. I just assume that we're being listened to all the time, either by Amazon or by the government, but it doesn't always act on it. In other countries, sometimes it does get acted on. So there have been cases in other countries of people being told, you were within 10 feet of someone with COVID-19, please self-quarantine, self-isolate, or get tested. So this is an interesting situation where we're really learning a lot about personal privacy, about leaving people to make choices or trusting governments to make choices for us. And it's just a very odd situation. But we can kind of look at the genetics of a pathogen, or we can look at those interpersonal connections, kind of see where something is coming from to learn more about it. When we're thinking about the spread of a particular pathogen, what we're looking at is whether someone actually has the disease. We're not just looking at mortality, we're looking at morbidity. So morbidity just means that you are in the state of having a disease. It's generally expressed without any reference to the population size, so it might be just total number of cases, and morbidity rate is measured as a percentage of population. So morbidity is just kind of a raw, gross number. Morbidity rate is standardized by a standard number, like for every 100,000 people, or as a percentage of the population. So in this picture on the right, it's showing the confirmed number of cases of COVID-19 in different parts of the world, the total number of deaths, and the mortality rate. So this is expressing, when it says the confirmed cases, that's expressing confirmed morbidity. And then the mortality part is showing mortality rate. So confirmed cases, again, is a measure of morbidity. One thing that I wanted to say is this image is from March 3rd, and it's showing 147 cases, so that morbidity measurement of COVID-19 in all of North America. As of today, so at 3 p.m. on Thursday, March 26th, there were 82,404 cases of COVID-19 in the United States, which is more than is in Italy or in China. So keep in mind that our population is quite a bit bigger than Italy's. It's not bigger than China's, but we kind of had we have different disease progress. So this SARS-CoV-2 has obviously existed in China for presumably longer than it has in North America, and specifically in the United States. So the fact that we have now surpassed them in cases is pretty significant, especially since our population is smaller than theirs. Again, when we're thinking about morbidity, that is different from mortality. Mortality is the state of being dead, so mortality rate is the number of people who are dying. There's a lot of different metrics of mortality rate. There's kind of like your likelihood within the population to die from this condition. There's also case fatality rate, which is if you get this particular virus or bacterium or pathogen, how likely are you to die? So there's different kind of ways of measuring that. You know, right now, this is kind of challenging to talk about because we're not just talking about it in the abstract, right? You know, we are putting our lives in danger. A lot of you are healthcare workers. You go out, you interact with people, and you don't really have a choice about it. You or your partners are healthcare workers, and you worry about your children and your parents. So I want you to be familiar with these metrics, but again, I recognize that this might be a really hard and not abstract thing to talk about right now. So when we're thinking about what determines morbidity, we actually look at two different factors. We look at incidence and prevalence. These seem kind of similar, but they're actually pretty different. Incidence does feed into prevalence. So incidence is the number or proportion of specifically new cases per unit time. So basically, how quickly is this spreading? It's just taking into account new cases. With prevalence, that's the number or proportion of total cases in a population at a specific time. So that's an indicator of how widespread the disease is. It takes into account both old and new cases. So to kind of visualize that a little bit better, this is HIV prevalence and incidence in the United States from 1980 to 2010. So you can see in the 80s, there were a lot of new HIV infections happening, but as we started to learn more about how HIV spreads, how to prevent it, and how to treat it, those numbers dropped. So incidence has remained fairly low, it's not necessarily declining too much anymore, but it's remaining pretty consistently low, but prevalence increases consistently. So prevalence, whenever there's new cases, even if the incidence is low, it's still reflecting new cases. So the overall prevalence is continuing to grow. Basically, if that incidence is anything above 0, prevalence will continue to increase, especially for something like HIV, where it becomes kind of a more latent or chronic infection that is managed. For something like Ebola or a prion disease, where there is a really high mortality rate, there might be kind of more of a balance in incidence and prevalence, because when someone has that condition, they are eventually going to leave the population by succumbing to that condition. So when we're thinking about incidents specifically, how many new cases are we getting? We can look at various patterns of that incidence. If something is sporadic, if it has a sporadic pattern of incidents, that means that it happens occasionally. There's no geographical constraint. It's just kind of random who is getting this condition and where. So one example, which is shown here, is tetanus. It used to be much more common, but now because we understand proper wound hygiene, especially with puncture wounds, we have pre and post-exposure prophylaxis. We're able to kind of get vaccinations for it and deal with it after the fact as well. We know that it's a risk factor, stepping on rusty nails. There's a lot of people who are able to avoid having tetanus as an actual medical condition. So it's kind of random where it happens. If there's a location where it's much more common or people are not practicing good wound hygiene, it might be a little bit, we might have these spikes of incidents. It's just really sporadic. The bad thing now, though, is that there's a lot of people who don't trust vaccinations, and so there's increasingly numbers of tetanus where there really shouldn't be, and especially in the United States, because we don't have to deal with it that much. When people do get tetanus because they don't have the proper vaccinations, their parents didn't vaccinate them, we have to actually confer with people in various other countries where it is still common with dramatically different healthcare systems than ours, because it's so rare for it to happen in our healthcare systems. So that one is an example of a sporadic disease in terms of patterns of incidence. Another pattern of incidence is endemic. So that means that something is consistently found or constantly found in a given location. This heat map of the world is showing cases of malaria or incidence of malaria. So if you look at the United States, malaria is not endemic to the United States. We would not expect to find any new cases of it there unless someone is contracting malaria somewhere else and then coming back to the United States. Malaria is endemic to parts of South America, Africa, and South Asia. If you're traveling to somewhere like India, Pakistan, parts of Africa, Southeast Asia, or specifically the kind of central or northern part of South America, it's really important to take anti-malarial drugs preventatively so that if you are exposed to it, you're less likely to contract malaria, which can be very severe. So that's kind of the good thing about an endemic disease is we know to look out for it. I think I've mentioned coccidiomycoides to you guys. That is a fungal infection. The spores are found in the soil. That is endemic to the Central Valley and to the Southwest United States. And so the tricky thing about some endemic diseases is in the valley, we might know about valley fever. We might know that it's a very serious problem for people who work in soil, work in agriculture and construction, or immunocompromised populations. But if someone goes into the ER with lung nodules and the doctor doesn't know, is not from the Central Valley, doesn't know that that might be a common problem, then maybe they don't have that knowledge of endemic diseases and it goes overlooked. So endemic diseases, we can kind of anticipate them if we know about them. If we don't have that knowledge, then you could be missing out on something by assuming it's something else. Another pattern of incidence is an epidemic. So again, remember that's a higher than expected number of cases in a short time in a very specific location. This plot is showing the percentage of deaths. So it's not necessarily showing incidence, but it's showing mortality. And so this is showing influenza and pneumonia associated with influenza. And remember that the flu is seasonal. There's times where we would expect there to be much more incidence and much more mortality from influenza, specifically in colder months, dropping down in the warmer months. So there's kind of a seasonal threshold. If it goes past a certain amount, if mortality surpasses a specific epidemic threshold, that's when we start being concerned and calling it an epidemic. So every winter, it's not that we have a flu epidemic, it's just a seasonal flu, but during some cases, some years, there have been actual influenza epidemics. So for example, in 2004, we see that peak right there. In 2005, there was another one, and then in 2008, that's where we had that H1N1 that was pretty serious and scared a lot of people. So we can characterize different diseases based on their incidence patterns, but there's a few other ways we can characterize those diseases once they're in individual human hosts. So when we're trying to figure out what the actual causative agent is, we use a process called etiology. We've talked before about Koch's postulates. These help us determine what the actual etiological agent is. So we think about microbes as being the causative agents of disease rather than simply being symptomatic of the disease. So when we're thinking about Koch's postulates, one requirement is that the suspected causative agent has to be found only in sick organisms, not in healthy organisms. So in this example, they're looking at blood samples from a healthy, cute little mouse and a diseased mouse. And the suspected etiological agent, the causative agent, is only found in the diseased organism. You also have to be able to isolate it and culture it. So this is tricky, and in the next slide, we'll talk about some exceptions to Koch's postulates. But for example, when people first started studying the soil of Antarctica, they thought that it was completely sterile. They thought that there was no bacteria present. Now we know that there are incredibly complex bacterial and algal communities in Antarctica. The tricky thing is that we just didn't know how to culture them. So this is kind of based on the assumption that we can culture things. Generally, our culturing mechanisms are set up to mimic the human or animal body. So oftentimes we can grow things in culture if we have the right plate, but it is a little bit tricky. So you have to be able to isolate it. That's also tricky because sometimes microbes rely on symbioses and they don't do so well on their own. And sometimes the symbiosis is with the host, sometimes it's with other bacteria. So you also have to be able to take that pure culture, inject it into a healthy organism, and cause the same symptomology, the same disease pathology. And then you have to be able to isolate it again. So in cases like strep and staph infections, this might be fairly straightforward. There are a lot of exceptions to Koch's postulates. So for example, some organisms really can't be cultured easily. Syphilis grows inside of cells, so we have to use cell culture even though it's a bacterium. Leprosy, we talked about how it grows on armadillos, so we have to use them basically to grow up leprosy in the lab. That's Mycobacterium is the genus for that one. And then Legionnaire's disease causes that pneumonia. It's caused by Legionella, but that basically you have to use lung biopsies in order to grow it. So some organisms can't be cultured easily. Again, that makes it hard to isolate them in culture. And then some symptoms can be caused by many types of different microorganisms, and some microorganisms can cause many different symptoms. So the same microbiological, I think is the term I was trying to think of. I don't know where I was going with that. The same microbe, when it's causing an infection in different people or in different locations within their bodies, might present different symptomology depending on how it interacts with their immune system, different things like that. So that makes it challenging if you were to isolate the culture and then inject it into a healthy host, to really know for sure that that's going to result in the same disease. And then we also have to consider the ethical considerations of host range. For example, HIV only infects humans. So if we were to try to use Koch's postulates, we would have to inject healthy people with HIV, which again is like a huge ethical consideration. So there have to be other ways of characterizing a particular disease, of identifying that infection and confirming it other than Koch's postulates. One is by looking at the evidence of how it's affecting the patient. Signs and symptoms are very similar, but they are pretty different in a lot of ways too. Signs are objective. They are a change that a clinician can see and measure. They can observe it. So that might be a lesion in the skin. It could be a rash. It could be a fever that you can measure. It could be paralysis. You can see whether a patient reacts to something, do a neurological test. So there's lots of different things that you as clinicians can observe. A symptom is a subjective change. It's something like a patient's pain level or general feelings of malaise. For example, in the earlier example of the rash, which is a sign, the itchiness of that rash, how it makes the patient feel, is a symptom. And then a syndrome is when you have a consistent grouping of signs and symptoms that accompany a disease. We can also characterize different diseases based on their rate of spread. So if something is communicable, it does spread directly or indirectly to another host from an original host. If something is contagious, that means that it is very communicable. So things like chickenpox are super contagious. Something that is non-communicable does not spread from host to host. So for example, tetanus. You can't spread tetanus to someone else if you have tetanus. You have to actually get those endospores through that parenteral route, and they have to be, put into your body a certain way. You can't just spread it from person to person. Something like cancer, for example, is non-communicable. We can also start to look at different patterns of transmission. So not just the rates of incidence, but really looking at how that transmission occurs. We broadly distinguish this by common source spread and propagated spread. With common source spread, it can be point source, continuous, or intermittent. With common source, what we're talking about here is that there is a single source of this particular pathogen for all of the individuals infected. So for example, with point source, that might be like food poisoning. If you have contaminated food at a family party, that's a common source that is there for a short amount of time. So point source, it is a point in time. It's a small, short time period where everyone is exposed to this particular reservoir of the infectious agent. If it's continuous, that means that it's coming from that common source over an extended period of time. So for example, if the city's water gets contaminated with sewage, that is going to continuously be exposed to different people. So that is a common source, but it's continuous. Then with intermittent, infections start and stop with various stimuli. So for example, if there's a lot of rainfall and that spreads sewage around and causes an infection, it's a common source, but it will stop when the rain stops. Then when the rain starts up again, sewage kind of rises and mixes with water and again will spread. So that is again a common source, but it's intermittent. So when we have common source, we have that single source, we distinguish it based on how often and for how long people are exposed to that common source. With propagated spread, that's person-to-person transmission. The person who gets the pathogen then transmits it to someone else, so each successive person becomes a source. With different patterns of transmission, so kind of continuing along that idea of categorizing based on patterns of transmission, the type of transmission actually determines how long the outbreak lasts, the duration. So for example, if it's point source, there's not going to be continual family parties where your rice has bacillus and you're all getting an infection as a result, right? It's just kind of like an isolated incident. And so with this particular one in the bottom left, it's showing disease incidence with point source spread. It's just kind of sporadic. It's not happening consistently. And it's maybe decreasing over time as you figure out where this is coming from. With propagated spread, where each person becomes a source and there's person-to-person transmission, you see it kind of increasing steadily over time, maybe going through these waves, but it gets kind of a lot worse before it gets better because it's just continuing and propagating. So that's what we're seeing right now with COVID-19. So in order for there to be this propagation and continuation of a particular causative agent, you have to have reservoirs. Some of you might have heard of Typhoid Mary. She was a woman who had asymptomatic typhoid fever. She did not have any symptoms of it, but she was full of it and shedding it and passing it on to other people. So a lot of cases of typhoid fever were because of her. Different animals can be reservoirs, animals that we cohabitate with or animals that we get exposed to or eat. With the current COVID-19 case, it was thought that the pangolin, which is that cute little guy on the bottom left, and the different bats, like those cute little flying foxes over there, are reservoirs of COVID-19. A lot of viruses come from bats actually, and because bats move around quite a bit, they're the only genuinely flying mammal, they're able to kind of spread things to different livestock. They also are eaten in some parts of the world, so they get exposed to us and they have a lot of motility in terms of how they move different pathogens around. So that spread of an infectious disease really relies on having one of these continual sources of the disease organism, or the causative agent. We'll talk about how we categorize those in just a moment. The source has to provide the pathogen with enough resources for them to survive and multiply. So that pathogen has to be able to hijack the host cells or the reservoir cells, or else survive in or on it. And it also has to have the opportunity for transmission. When we're thinking about different reservoirs, we can categorize them as living or non-living. I'll briefly talk about non-living first. We've talked about this already, so those are just fomites, and then we'll get back to living. So a non-living reservoir of a pathogen is a fomite. We briefly talked about this in week 4. When we're thinking about fomites and how likely they are to act as reservoirs and spread pathogens to humans, we think about how closely it's associated with human tissue. So for example, an electrical wire outside, most of us aren't going to be touching those. They are probably not going to act as fomites. But something that is like a surgical implant or your phone that you come into contact with a lot, that is very closely associated with human tissue. And also, if the substrate itself is more likely to harbor the right environmental conditions to grow that pathogen, then it's going to really act as this non-living reservoir of disease. So for example, a sponge that is just sitting near the kitchen sink, it's damp, it can get warm. That is an excellent fomite for pathogens to grow. So when we're thinking about living reservoirs, we can divide them into passive carriers or active carriers. In these cases, these are both humans. So we're going to focus on human living carriers. If they're passive, we don't consider them to actually be infected. They're contaminated, but they're not infected. So that is something like transient flora. If you are like walking around, you put your hand down, maybe you're taking public transportation, or you're otherwise coming into contact with a lot of people, you're picking your kids up from daycare. If you just touch something briefly, there might be a lot of bacteria, a lot of viruses, a lot of fungal spores just sitting on the surface of your hand. If you wash it correctly with those CDC hand washing procedures we're all becoming very familiar with, then those usually get washed off. So this is just passive. You're not actually infected. You're not actively shedding it. You just kind of have this sitting on your surface. At first, with COVID-19, people were very scared that dogs and cats were able to carry it, especially since a dog tested positive for it. But it turned out that they had just come into contact with their human who was infected, and they weren't actively growing or shedding the virus. If there is an active human carrier, they are infected. So maybe they're sneezing, maybe they're, sending out the pathogen in some other way, and they can continually shed and transmit the pathogen. When we're thinking about that active infection, in the previous image with Brian Cranston, that was what we call symptomatic. So symptomatic carriers pass on the infection when they are showing signs and symptoms. Typhoid Mary was an example of an asymptomatic carrier. They maybe pass on the infection before or after they show the symptoms, or they don't show any symptoms at all. This plot in the bottom right to me is a little bit hard to see, but to me it is wild because what it's showing, the red bars are rates or prevalence of COVID-19 cases in different age groups. So that one that's really high, or the red bars are showing in South Korea, where there's a lot of testing for COVID-19. The green bars are showing in Italy, where they only test people with symptoms. So you can see that they track fairly well that people who are older adults are more likely to show severe symptoms. And so that's why I think the green bars are really high in those older age brackets in Italy, because those are the people who have symptoms. They're actually getting treatment for it. Whereas in South Korea, you can see this huge bar for, I think it's 20 to 29-year-olds. So these people are very much infected. They're asymptomatic. They aren't showing symptoms, but they can for sure pass on that infection to other people. So just because you yourself feel healthy and are not showing symptoms doesn't mean you should be walking around studying in groups, not practicing social distancing. So just because you feel good doesn't mean you're not actively spreading this to people who may die as a result of it. So those were human examples of living carriers. There are also non-human examples, which are zoonotic carriers. And again, those might be symptomatic or asymptomatic. With swine flu, that does cause symptoms in both pigs and humans. So not just H1N1, but all swine flus affect both pigs and humans. For hantavirus, which you might have heard of, you might be doing that for your disease project. Those cause very serious disease in humans, but they don't cause disease in the mice that carry it. So when there's a lot of rain, that leads to more vegetation. More vegetation leads to more mice, which leads to more hantavirus. People go out and sweep up kind of dirt that collects. They might sweep up mouse poop, and then the virus actually gets into your respiratory system and causes a very severe disease, hantavirus. So it's asymptomatic in the mice that carry it, but it's very symptomatic in us. So these are both examples of zoonoses. Zoo means animal, and so these are different infections, different conditions that are passed on by animals to humans. So when we do have a reservoir and we also have a host, the pathogen has to get from the reservoir to the host. So that can happen through direct or indirect contact. It can happen using vehicle transmission. That's when the pathogen is transmitted through vehicles like water, food, or air. So those might also be referred to as different media. It's basically like hopping in a vehicle and traveling through something like water or food or air to get from the reservoir to the host. There's also vector transmission, where the vector is an animal like a fly or mosquito that carries the pathogen between two different hosts. So that vector is not necessarily a host itself, it's just traveling between a reservoir and a host, or between two hosts. So with direct or indirect contact transmission, direct contact or person-to-person contact might be congenital, or it might be person-to-person through like touch and interaction, or different bodily fluids. It might be from mother to fetus or mother to baby through congenital or vertical direct contact transmission. There's also horizontal direct contact transmission. So again, like shaking hands with your friend or high-fiving them or being too close when you're studying for an exam. With fomites, again, those are non-living, so this is indirect contact transmission. I have the flu, I cough on my hand, I put my hand down on the table, flu virus ends up there, someone else comes by, touches that same spot, picks up the flu virus, rubs their eye or puts their finger in their mouth, and then they have the flu. So that's indirect contact transmission using fomites. There's also droplet transmission. So to me, this picture is wild. It's showing you all of the droplets that come out of your mouth when you sneeze. So this is why it is so important to sneeze not into your hands and definitely not into the surrounding environment, but into your elbow or the crook of your arm so that it's completely covered up. Because when you sneeze, so many droplets come out. Remember, this is also why you should never flush a toilet with the cover or the lid open. It's not just a matter of whether the seat is up or down. You have to have that whole sucker covered so that when you flush that toilet, all of that bacteria does not become aerosolized and travel through droplet transmission throughout your entire bathroom and land on your toothbrush. Vehicle transmission, like I mentioned, moves that pathogen through different media. So for example, it might move through the air, or it might be waterborne or foodborne. Airborne, you might think about things like spores, fungal spores. If a fungus releases those spores, they can travel through the air. Airborne is kind of related to droplet transmission, depending on whether the droplets are coming into direct contact with us or whether they're traveling through the air to get to us. Waterborne and foodborne are closely related. The images on the right are showing Giardia lamblia. That one is a protozoan which is able to form a cyst. The cyst ends up in water or food. It is reinforced, so it makes its way through our digestive system, and then once it gets down into our gut, it becomes that active trophozoite stage again, and so it's able to cause pretty severe disease. With foodborne transmission, we always want to make sure that we're being careful of cross-contamination. Never use the same cutting board for like raw meats versus vegetables and bread, unless you're really thoroughly scrubbing it in between. And even then, it's usually best to just use separate cutting boards. There's also the fecal-oral route, so that, a relationship between getting feces into your food and it ending up back into your digestive system. That is how a lot of different pathogens are spread, just through like poor hygiene, not washing hands, or sometimes if you are really reliant on, for example, farming of rice, where people might have fecal material on their clothing and they're submerged in this water that is then getting onto our food. So there's a lot of different ways the fecal-oral route can happen. With vectors, those can be mechanical, like a housefly. By that we mean that maybe the fly is flying around, it physically lands on your food, it gets some of that, maybe it has landed on fecal matter before, it has that fecal matter still sticking on its feet, and then it puts that onto your food, and so it is physically, mechanically moving that feces to your food and completing that fecal-oral route. So then you're getting contaminated food. That's part of why it's, we really freak out when flies get on our food, because we have this innate knowledge that they are mechanical vectors for pathogens. Vectors can also be biological. Mosquitoes are one of the most common examples. If they are physically interacting with your fluids by biting you and getting exposed to your blood and moving that blood to someone else, they can spread very severe pathogens. And that's how Plasmodium, the protozoan that causes malaria, is spread. So, parasites can also have very complex life cycles where they have lots of different vectors and hosts. If they have an intermediate host, like for example with this tapeworm, uses crustaceans or small fishes or different things like that as its intermediate hosts. And then those are situations in which in its life cycle it is still considered immature. If the parasite is in its preferred host and it is fully mature, that's called the definitive host. So one thing that I wanted to touch base on is why quarantines are so important. I'm sure a lot of you have seen all the stuff about flatten the curve, making sure that we don't overwhelm healthcare systems. So when we quarantine, what we're doing is isolating people who may be passively or actively carrying a pathogen. So quarantine is technically different from social distancing or social isolation, or self-isolation. Social distancing is just general best practices to make sure that we are minimizing the spread of a condition because we don't know for sure if we're passively or actively carrying it. Self-isolation is when you think there's a good chance you've been exposed to something, but you aren't showing symptoms. A lot of healthcare professionals right now are self-isolating. So this morning on NPR, there was this story about an epidemiologist. She just had a baby, so she has an infant, and she has several other children. Her husband is a doctor, and he's constantly being exposed to coronavirus, to patients with COVID-19. So he's basically living in a completely separate part of the house from them. He's in self-isolation so that he does not spread that virus, which he probably has picked up, to his brand new baby. So it's, self-isolation is when you think you've been exposed, but you're not showing symptoms. Quarantine, people are often showing symptoms, but it might be isolating people who are passively or actively carrying this particular pathogen. So for example, with all those cruise ships, those were in quarantine because they were probably exposed to the pathogen. It's hard to predict the spread of disease on a cruise ship because that air is circulating, so people are spreading the pathogen between different cabins. So it was not great for the people on the ship because it spread super quickly through the ship. It was helpful for maybe preventing those people from spreading it to other people on the mainland. So that quarantine really depends on who you're trying to help. Again, we know that this might be in a clinical setting or it could be at home like we're experiencing now. And the duration of that quarantine entirely depends on the incubation period, the evidence for infection, the presence of different signs and symptoms, as well as other metrics of epidemiology. We care about this as people who are interested in healthcare because this idea of healthcare-associated infections or nosocomial infections really relates to how diseases spread in hospitals and whether best practices are being followed. So hospital settings have a lot of compromised hosts. They have this massive chain of transmission between staff and patients or fomites, and there's plenty of microorganisms that are found in a healthcare environment when people are coming in with infections. It's so important to follow best practices and standard precautions. Right now, we are running out of a lot of basic healthcare supplies, so people can't use PPE the way they're supposed to, but you just got to do it to the best of your ability. Even in the best of times, according to the CDC, healthcare workers only wash their hands 40% of the time before interacting with patients. So please make sure you are doing better than the people who came before you. In the next lecture, we'll talk a lot more about how a particular pathogen ends up getting inside of a host, really colonizing it and adhering to it, integrating into those tissues and evading host defenses. So we'll think about the progress of an infection in that context. Here we're going to think about how it relates to transmission. So when a bacterium or another pathogen infects a host, how does the progress of its infection actually allow the host to shed that particular pathogen and transmit it to other people? Again, looking at the epidemiological aspect. So this plot has what are called the 5 periods of disease. These are also called stages or phases of disease. But when we're looking at this, this is basically the common sequence that diseases follow in terms of how they colonize and grow within a host, and also the emergence of different signs and symptoms and how contagious or transmissible it might be. So this first period or stage or phase, whatever you want to call it, is the incubation period. That's the time that it takes for there or the time in between the host first becoming infected with the particular pathogen and then first displaying symptoms. So that's signs or symptoms of any sort. It doesn't have to be unique or special for this particular infection. It's just in between actually getting infected and when you know that there's something wrong. So it can be consistent or it can be variable. It can be contagious or not contagious during this point in time, transmissible or not transmissible. So it's just kind of a tricky time. The pathogen is colonizing the host and it can do a lot of different things. When the signs and symptoms first start emerging, that's called the prodromal period. So it occurs in some diseases. It doesn't happen in all diseases. It includes kind of early, milder symptoms like different aches and malaise. A very common example of this is with HIV. So when someone has HIV, they usually get this prodromal period where it feels like they have the flu. They feel kind of achy. They maybe have some respiratory issues, and then it kind of goes into latency and builds up the viral load over time if you're not actively being tested or treated for it. So that prodromal period really stands out when you have HIV infections. In a lot of different cases, it's just this like common achy feeling, period of malaise, so not unique symptoms. It may mimic other syndromes, it may mimic other diseases. So at that point, you know something is wrong, you know that you might be contagious, you might be able to transmit this particular pathogen, but you maybe don't quite have enough information to know what that is. Then there's the period of illness. This is kind of getting into what's also called the invasive phase. This is where you have some of those really unique signs and symptoms that help you diagnose what might be causing this. Your immune system is activated. At this point, the patient is either going to overcome the infection or they're going to succumb to it. So what this plot is showing us, I should probably explain that. On the x-axis is time. On the y-axis is the number of infectious agents. And you can see that this kind of follows a bacterial growth curve. There's kind of that lag phase of it growing and getting used to things, exponential growth, and then kind of a balancing off. At this point, what we call the acme, this is the highest number of infectious agents. That bacterium or virus is going to drop off either because the host has started fighting it and their immune system is able to overcome it, or because the host dies and so there's no host for those infectious agents to continue replicating inside of. So either way, we get into the decline phase, which still can be contagious. It's not always contagious. At this point, those signs and symptoms are starting to drop down. There might be still some immune system activity like fevers, but they might be starting to decrease. At this point, though, because the patient has gone through this serious immune response, because the patient has been exposed to a lot of different things, they're kind of vulnerable. They might be vulnerable to secondary infection with a different opportunistic pathogen. Convalescence period also can be contagious, but it is not always contagious. So there's a lot of cases in which it's not. At this point, the patient is kind of getting back their strength, really clearing the last traces of that infection from their body, but the disease is no longer really affecting their bodily function. So they're in convalescence, they're kind of healing and recovering. We again already took the lecture exam. I'm late to record this, but please make sure that you are staying up to date with everything that's posted to Canvas on both the lecture and lab pages. I'll do a better job of reminding this to you guys, but you really need to make sure you're posting on those discussion boards for both lecture and lab. Since you guys don't get the opportunity to even meet up at a Starbucks and study with each other, you really have to do a lot of the opting in to some of those online learning spaces. So please make sure that you're using Zoom, that you're using Canvas. If you don't have access to those, there are a lot of workarounds, and I really encourage you to try to get to campus by Friday the 27th, which is basically today, when I'll be posting this. But it's really important for you to get some of those free resources. There's free laptops, there's free hotspots. If you don't have reliable internet, Comcast is giving 2 months of free internet depending on your income. So there's a lot of different ways we can get you involved in the class. You don't have to give up. We're really trying to be here to support you. So please make sure you're checking those resources and staying up to date on them. Thank you.